Welcome to Educon. My name is Imam, and today in this video we'll discuss about endomembrane system. Endomembrane system consists of endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Why they are included in endomembrane system? Because their functions are coordinated. Their functions are coordinated. Since the functions of mitochondria, chloroplast, and peroxisomes are not coordinated with the above components. Above components here means above organelles. These are not considered as part of the endomembrane system because mitochondria and chloroplast and also peroxisome, their functions are not coordinated with the ER, Golgi complex, lysosomes, and vacuoles. Hence, they do not come under endomembrane system. Coming to endoplasmic reticulum, you can see the image on the right side. Electron microscopic studies of eukaryotic cells reveal the presence of network of reticulum, network or reticulum of tiny tubular structures scattered in the cytoplasm. Okay. Now here, this is also important. Where what? Network or reticulum of tiny tubular structures. Okay, that is which is called as endoplasmic reticulum. And endoplasmic reticulum divides the intracellular space into two distinctive compartments. One is the extraluminal, which is called cytoplasm. Okay. The other one is inside the endoplasmic reticulum, which is called luminal space. Okay. And sometimes endoplasmic reticulum often shows ribosomes attached to its surface on the outer side. So if the ribosomes are present on its surface, then it is called rough endoplasmic reticulum. On the side you have, this is also a endoplasmic reticulum, but they do not have any kind of ribosomes attached to its surface. Hence they are called smooth endoplasmic reticulum. And rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis and secretion. So here, because ribosomes are associated with the protein synthesis, hence the rough endoplasmic reticulum is frequently observed, means it observed in majority it is observed in cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. You can see here, this is the nucleus and this is the membrane of the nucleus, the outer membrane, and from here endoplasmic reticulum, they are in continuous with the outer membrane of the nuclear membrane. Okay, the smooth endoplasmic reticulum is the major site for the synthesis of lipid, and in animal cells lipid like steroidal hormones are synthesized in smooth endoplasmic reticulum. So here we have certain points to remember. Okay, like they are in the form of network or reticulum of tiny tubular structures. Okay, whereas smooth endoplasmic reticulum, they are also same, but they do not have any kind of ribosomes on its surfaces. And rough endoplasmic reticulum is involved in protein synthesis and also secretion. Okay, you need to remember these two things. And when it comes to smooth endoplasmic reticulum, it is the major site for the lipids. And the most important thing to consider here is steroidal hormones in animal cells are synthesized by smooth endoplasmic reticulum, because hormones, okay, these steroidal hormones, okay, lipid, they are like lipids. Okay, so we conclude this and we go to next organelle called Golgi complex. Camillo Golgi, 1898, first observed densely stained reticular structures near nucleus. So they are, before we come, network or reticulum like tiny tubular structures. Okay, here reticular structures near the nucleus. Okay, they were later named as Golgi, named Golgi bodies after Camillo Golgi. They mainly consist of flat disc-shaped sacs or cisternae of 0.5 millimicron to 1 millimicron in diameter. Okay, these are stacked, and these cisternae, they are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. So here varied number, because there is no any specific number of cisternae like 2 or 10 or 15 like that. So it varies from cell to cell. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cysts or the forming phase. I will tell you about this convex and convex cysts or the forming phase. And concave trans are the maturing phase. The cis and trans phases of the organelle are entirely different but interconnected. Okay, so here now you can see here, this is the cis, convex cis or forming phase. This is the maturing phase, from where the vesicles move away from the, after being prepared here, because from endoplasmic reticulum, the material gathered, they come and join near the cis phase, and they move towards the cisternae. As the cisternae, they are connected in the bottom. Okay, it is a, we are not seeing any kind of connection between the cisternae from top, but they are connected, and from there the packaging material which comes from the endoplasmic reticulum, they move towards the cisternae and exit from the maturing phase. Okay, the cis phase and trans phase of the organelle are entirely different but are interconnected. Okay, the Golgi apparatus principally perform the function of packaging material. Remember this. Main function of what the Golgi complex is, it packaging materials to be delivered either to the intracellular targets or secreted outside the cell. So the delivered stuff will be released into the cytoplasm, from where it will reach to intracellular targets or going outside the cell, secreted outside the cell. The materials to be packed in the form of vesicles from the ER fuse with the cis phase of the Golgi complex. As I told you, the materials which comes from the endoplasmic reticulum fuse with the cis phase of the Golgi complex and move towards the maturing phase. This explains why the Golgi complex apparatus remains in close association with the endoplasmic reticulum. Okay, now you understand why they come under endomembrane system. Their functions are coordinated. Hence here the materials should be packed from the ER, fused with the cis phase and move towards the maturing phase. From there it is released into the intracellular space or cytoplasm and deliver either directly to the intracellular targets or secreted outside the cell. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the Golgi complex before they are released from the trans phase. Okay, see here the number of proteins synthesized by, because proteins are synthesized by ribosomes. Okay, ribosomes are present on endoplasmic reticulum. So these proteins are modified. Remember this, modified in the cisternae of the Golgi apparatus. As we see lot of cisternae parallelly stacked together. So these packaging, this material which has to be packed, they move, the proteins, they move from one cisternae to other cisternae before they are released from its trans phase. Okay, here what happened in this process, the proteins are modified in the cisternae. Remember this, proteins are modified in the cisternae. Golgi apparatus is the important site for the formation of glycoproteins and glycolipids. Okay, so what is Golgi complex? It is named after Camillo Golgi. Okay, they have parallel sac-like structures called cisternae. Cisternae are connected together and it is concentrically arranged near the nucleus. The materials to be packed from endoplasmic reticulum fuse with the cis phase. This is the cis phase, this is the trans phase. From here the materials move along the cisternae and towards the maturing phase before and release from the maturing phase. Now you understand the process. And also Remember the thing, glycoproteins and glycolipids. Okay, they are for them, Golgi apparatus is the important site for their formation. Coming back to lysosome, these are membrane-bound vesicular structures formed by process of packaging in the Golgi apparatus. Lysosomes are membrane-bound vesicular structures. Okay, this is a single term we must remember. Okay, where it is formed? It is formed in the Golgi complex by the process of packaging. As I told you, the function of Golgi complex is packaging. Okay, the isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. So, what are the hydrolytic enzymes? Hydrolysis means lipases, proteases, and carbohydrates. Optimally, which are active at acidic pH. So, acidic pH means less than 7, you go 6, 5, 4. This is called acidic. If you go above 7, it is a basic. So, these enzymes, they are active at the acidic pH, and the enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acid. Carbohydrates, they digest the carbohydrates. Proteins by proteases, lipases, lipids. So in that context, so what we know about lysosomes here, they are membrane-bound vesicular structures formed by the process of packaging inside the Golgi complex. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, such as lipases, proteases, and carbohydrates, which are active at acidic pH. Okay, we have around 3 to 4 points which must be remembered. Coming to vacuoles. Vacuole is the membrane-bound space found in the cytoplasm. Okay, vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials which are not useful for the cell. And the vacuole is bound by a single membrane. Remember this, which is called tonoplast. Okay, it is a, what we learned, vacuole is a membrane-bound space found in the cytoplasm. It also contains water, sap, excretory product, and other materials which are not useful for the cell. And vacuole is bound by a single membrane called tonoplast. Okay, in plant cells, the vacuole occupies 90% of the volume of the cell, which is not the case in animal cells. In plants, tonoplast facilitate the transport of number of ions and other materials against concentration gradient. What is concentration gradients means? From higher concentration to lower concentration. We studied about the membrane transport of how the transport of molecules or ion takes place in plasma membrane. So there is a passive method and there is a active transport also, where energy is involved, where energy is not involved. Okay, so here the tonoplast, the membrane surrounding the vacuoles, facilitates the transport of number of ions and other materials against concentration gradient. So we know about the concentration gradient. Against means it involves it involves energy, because from lower concentration to higher concentration, movement of ions or molecules, any other material from lower concentration to higher concentration. This is against the concentration gradient into the vacuum. See, remember, concentration gradient is according to concentration gradient means moving of molecules from higher concentration to lower concentration. Same against concentration gradient means moving of molecules from lower concentration to higher concentration, where energy is involved, that is ATP. Here, hence their concentration is significantly higher in the vacuole than the cytoplasm. Yes, because now you understand here, because the transport is against the concentration gradient, so the concentration of the chemical, the materials, are higher in the vacuole than the cytoplasm, because the tonoplast facilitates the transport of ions against the concentration gradient. In amoeba, we see contractile vacuole, which is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. So these are the two other examples given in NCERT book. In an where you can see contractile vacuole, you can see it in amoeba. We need to remember this. What is the membrane surrounding the vacuole is the tonoplast, and food vacuoles in protists. Okay, protists food vacuoles are formed by engulfing the food particles. Okay, with this we conclude this endomembrane system. My request to all you guys, do one thing, after listening to the video, kindly go through the NCERT book and revise it as many times as possible. Okay, when you read continuously, you need to make a lot of revision to remember everything. Then only the questions will be easier for you. Thank you.